Looking forward to your talk.
Well, thank you very much, Katie, for that nice introduction. And I'll do my best to remember to mention something about that. So let me just click on that. So today I'm going to talk about 4D microscopy and how it enables us to obtain unique insights into immune cell function, cellular infection. So as Katie mentioned, I'm going to talk a bit about malaria, which is the main focus of my lab. We collaborate closely with a number of labs, but especially with Alan Cowman's lab. And I'll also mention a project that we undertook together with Kate MacArthur and Ben Kyle, looking at apoptosis. So I wanted to begin here, since I'm going to be talking sort of mostly about development of imaging, recent development of imaging techniques or applications thereof. I thought it would be appropriate to lead in with the following quote by Sydneybrenner, which comes from a lecture he gave around 1980. I'm not entirely sure if he continued to believe this towards the end of his career, but nevertheless, it is progress in science depends on the interplay of techniques, discoveries, and new ideas. So Sydneybrenner, as you know, received the Nobel Prize in Medicine in 2002 for his discovery concerning the genetic regulation of organ development and programmed cell death. And he was an early adopter of new techniques for cloning and sequencing DNA, and he made the decision to study C. elegans, which is a simple experimental system and ideal for things like looking at genetic programming and microscopy. So this really enabled him to look at link genes and their functions, in particular to things like cell division and organ formation. And one of the things he said during his Nobel Prize lecture was, if we take 2020 as a year of good vision, we may note that this is 17 years in the future. And it represents the same time today from 1985, when the first discussions began about sequencing the human genome. And you might recall that the human genome was mostly fully sequenced around 2003. And he believed that while it was important to identify and characterize gene loci, it was important to understand the actions of their products and how they are integrated into the physiology of cells. And that one of the most powerful ways to obtain this information was through the use of microscopy. So this brings me to this slide here, which I decided to put together because in fact, since 2010, since 2000, we've seen some big advances in light microscopy. And all of these images here that you see from left to right are images captured by either myself or people I was collaborating with. And I decided to put this slide together to show how microscopy has evolved over the last couple of decades. 20 years ago, for example, I don't know if you can see my mouse, but there was always this quite a large trade-off between wanting to have high spatial resolution or high temporal resolution. So it was very difficult to sort of have both because we just either didn't have the sensitivity at that time or we just didn't have the kind of techniques that we're able to work with today. So, I was really fortunate, as Katie already mentioned, to undertake my first postdoc at the Institut Pasteur in Paris, and that started in 2002. And I was collaborating closely with Spencer Short, who's from the UK originally, and he was running really one of the most cutting-edge imaging facilities in the world at that time, I think. And back in those days, the best resolution we could get was really from confocal coupled with deep convolution, which is sort of can see here. This is just a COS cell expressing GFP, a porin calcium reporter in mitochondria. But the interesting thing was back in those days, we even had a RAID array or a little mini server inside of the imaging facility. And we were using Huygens deconvolution software. And we would take a simple Z stack on a confocal. We were using the Zeiss LSN 510 at the time. And then we would take that file and pop it into Huygens deconvolution software, put it on in the afternoon and come back the next day to find that it had finished deconvolving. So, there's certainly not like the speed at which we can do things today, but this is really just to show you how things have evolved. So we worked a lot with bioluminescence at that time using a calcium reporter because, you know, we didn't have to use any light excitation. So these were, this was working on bipolar cells from the retina, where obviously light can stimulate those cells. And, you know, I moved to WEHI in 2009, where I established the Centre for Dynamic Imaging. And at that time, this is sort of the type of images we were able to obtain of malaria parasites. So these blue spots here are nuclei with the rock trees labeled with GFP. But if you look at what we've been able to obtain, for example, more recently, this is sort of the same protein now that you're looking at, but you can see that you can see this nice localization of the protein in the apical region of the parasite. So, you know, we've really come a long way. Here you see just resonance scanning confocal versus lattice light sheet, which I'm going to talk to you a little bit about. So it has evolved quite a lot. Very, very quickly, these are the many different types of biological processes that we typically study at WEHI as a medical research institute. And this is just to show you really the wide range of scales that we work across, not just spatially, but also temporally. So to summarize what I'm going to talk about today, my talk is sort of broken up into three parts. So the first part of my talk will be about malaria and the work that we've done on the blood stage of malaria, looking at how malaria parasites invade red blood cells. And then the second part will be telling you a little bit about another project that we were closely collaborating on, and that's looking at BAX-mediated apoptosis and release of mitochondrial DNA. And that's the work that we did at Genelia. And then if I can get to it on time, I'll cover very, very quickly just a couple of slides on work we did together with Danny Ratnayaki and Peter Curry from Monash University, looking at the role of macrophages in muscle regeneration. So I'll start here with malaria, and I'll quickly tell you a bit about malaria. You know, malaria, and in particular, the life cycle of malaria is shown here. So you see when a mosquito bites the human and injects this form of the parasite, which is known as sporozoites, they travel into the bloodstream, and from there they make their way to the liver, where they infect hepatocytes. And then during that time, they multiply inside of hepatocytes, and then they burst out. They enter into the bloodstream as merozoites, and these merozoites, which are only about 1 to 2 microns in size, then begin invading erythrocytes. And then when they invade the erythrocyte, they undergo a developmental program in there over about 48 hours, where they develop, and then they multiply again, and they then undergo egress, releasing about 16 to 20 of these merozoites into the bloodstream again, and the cycle continues. So this is sort of known as the asexual stage of malaria, and it's the blood stage of malaria. And this is the stage that we're very much interested in studying, as it's, you know, where a lot of focus has been to sort of find treatments or vaccines that are capable of blocking this stage of infection. So if a picture is worth 1,000 words, I think everyone understands that a video is really worth thousands more. So if you wanted to understand how to play basketball for the first time, you'd have difficulty learning the rules, especially if it was just, if you had to learn those rules just from snapshots alone. And even if you looked at the photos of thousands of games, you still wouldn't understand the rules. But if you had the opportunity to watch a few games live, you'd probably already have a pretty good grasp of the game. And then if you analysed each play carefully, where it is possible to see how each player behaves and interacts and what kind of outcomes these interactions cause, you'd quickly develop a pretty good understanding. So this is just to highlight that live imaging allows us to define some of the more complex rules of biology. So the first time Plasmodium falciparum, or Plasmodium, which is the species of the malaria parasite, was captured invading a red blood cell in a live time-lapse recording was in 1975. So it made the cover of Science, which you can see here, and this is actually the highest resolution image I could find. And, you know, at that time, it really gave, just being able to capture this event live gave a great deal of detail. You know, it was challenging because, you know, live microscopy of invasion is not easy because, as I already mentioned, the parasites are very small. The process of egress is quite stochastic in nature. And because the event is very fast and parasites are also very sensitive to light, this does in fact represent some pretty big challenges. So until recently, it's been difficult to track and quantitatively assess invasion in real time. And this means that it's prevented us from being able to answer some of the fundamental questions regarding what the sequence of events actually is, how the parasitophorous vacuole membrane is formed. So when the parasite invades into a red blood cell or becomes internalized, it becomes enclosed in what is known as a parasitophorous vacuole membrane, where it remains protected and where it grows and develops. And then it exports proteins out into the red cell, effectively setting up a little home for itself. And there it continues until it starts to multiply and then undergoes this cycle that I mentioned before. But, you know, some of the other things that we still don't understand is how is that parasitophorous vacuole membrane formed? And how does it seal after parasite internalization? And what role is actually played by the host cell? So these, back in sort of 2010 and 2011, I really had the great opportunity to collaborate closely with Dave Rigler and Jake Baum, who were both at the Imperial College of London. And their objective was to really deduce the sequence of events and some of the functions of the key ligand receptor interactions. And Dave and Jake came up with quite an innovative way to use synchronized malaria cultures and then fix them at different time points after parasite egress. And they also labeled, so here you see these are super resolution images that they subsequently captured of those different time points. And they labeled the tight junction. So the tight junction, you see there's this ring in green here. The tight junction is this kind of aperture that is formed by the parasite that the parasite pushes into as it becomes wrapped by the red blood cell membrane. And that aperture remains very tightly sealed against the edge of the parasite. And they used this protein RON4, this labeled RON4, in order to be able to determine what stage of invasion they were actually looking at. So this enabled them to look at early stages of invasion, mid-stage invasion when the ring was fully enclosed around the parasite. So in blue is the nucleus of the parasite. In red is a marker that is found in the apical region, which then is released onto the surface of the parasite. And then here you can see where the parasite has fully entered into the red cell and where the tight junction is kind of closing up behind the parasite after it has invaded. And as these were sort of some of the first super resolution images captured, it happened to be the first super resolution microscope that was installed in Australia. And it was based at UTS in Sydney, and it was run by Cynthia Whitchurch and Lynn Turnbull. And below you can see that they also showed these electron microscopy images to correlate to, because up until then, that kind of high resolution hadn't been obtained, and it was important for them to do that. So they also looked at a couple of different inhibitors, including R1 peptide, which blocks tight junction formation, and they showed that when you block the formation of the tight junction, then the proteins or the contents of the rock trees, which are these apical organelles that are critical for invasion, those contents get released onto the surface of the red cell. But if you if you don't block tight junction formation, but you block the actin-myosin motor, this is the motor that's used by the parasite to drive into the red cell, then the ruptury contents do get released into the red cell, but the parasite is blocked from invasion. So they really, using this approach, were able to deduce some really important parts of the sequence. So then a team in Melbourne, actually, at the Burnett, led by Paul Gilson and Brendan Crabb, and they then developed this live cell imaging assay using differential interference contrast, looking at, and you can see here what we saw. I'll just show you again. This is egress of an infected red blood cell. The merozoites pop out. They interact with the red cell membrane. When they do, they cause these deformations on the membrane. So they're kind of pulling, and then eventually you'll see the parasite becomes internalized. So this was the best way at that time to really image this process, because as I mentioned before, you know, these parasites were quite sensitive to light, and light, you know, was really quite toxic. And they came up with this schematic in doing these experiments to show sort of the sequence of events that they understood up till then. One of the things they identified too for the first time was this calcium flux, which occurs right at the beginning of internalization. So when the parasite interacts with the membrane, there's a ligand receptor, an essential ligand receptor interaction that takes place. This leads to a calcium flux, which they identified at the apical region of the parasite rather than inside the red cell, which is interesting because they loaded red cells with Flo4 and not the parasite. So they actually proposed at that time that there's probably a pore formed in the membrane and that Flo4 probably travels back up into the apical region of the parasite where there's this calcium flux which occurs. So just this will show you now here the calcium flux that I'm talking about. So these red cells are loaded with Flo4 because you remember that while, you know, from my experience, I've worked with many different calcium reporters over the years. And honestly, the best that I've worked with up to now is probably GCAMP 6. I haven't had an opportunity to use GCAMP 7, but it's a genetically encoded calcium reporter. And obviously we can't use that in red cells because they're really just a membrane, you know, a sack of, you know, buffers and things, and there's no capacity to transcribe any proteins there. So we use Flo4, and you can see that, you know, from blue to red shows you the different concentrations of calcium. But again, you see the merozoite interacting with the membrane, pulling on the membrane, then bang, you'll see this very bright calcium flux, this sort of microdomain of calcium at the point of entry, and then the parasite invades. And this just shows you a still shot of a merozoite that is invading a red blood cell. So in gray, here's a red blood cell membrane. Here is the merozoite with one of those proteins that was identified as being essential. So there were these 3 proteins from the parasite that are released in a sequential manner. They form a complex, they bind to the basogen receptor on the red blood cell membrane, and this triggers the calcium flux, which then leads to the formation of the tight junction. It's not yet established if they're linked, but it is known that the tight junction is formed after that. And then at that point, the parasite uses its actin-myosin motor to drive its way into the cell. So we also tried to take it a sort of a step further, and we decided to use resonance scanning confocal. And some of you may say, why did you use a spinning disc? We actually don't have a spinning disc, but we have this great, you know, Leica SP8 resonance scanning confocal, which we've found we can get quite good results usually with live cell imaging. But here we just do very fast Z stacks And we were able to visualize quite effectively the malaria parasites invading red cells, and we were also able to detect the calcium flux. I will point out that, interestingly, that when Paul Gilson and his team were doing these calcium flux experiments, they only identified the calcium flux in about 50 to 60% of the cases of invasion. And they weren't really sure at that time exactly why they didn't identify the calcium flux in 100% of the cases. So there were some questions around whether this was actually an essential function for invasion. When we were doing resonance scanning confocal, we identified it in about 80% of cases, and in those, or maybe more, and in those cases where we didn't, we felt that it was only because we didn't drive the Z stack far enough beyond each end of the cell. So we probably missed it. So we sort of expected that if we had a more extended Z stack, and by the way, that was heavily undersampled because we didn't want to be too toxic. So we used the resonance scanner, which does tend to be a little bit less, much less phototoxic than a conventional scanner. And we just undersampled in order to make sure that we could capture the events. So I'll just show that to you one more time. So you can see laterally, we get quite good resolution. The data is very noisy. And then as soon as it turns on its side and you look at the cells in the axial direction, they kind of, like Niall always describes them as cinnamon scrolls. But you can see that the resolution is not very good. So, you know, we really were, really wanted something that would give us much better spatial temporal resolution. So when I saw, you know, after, you know, since I had been doing live imaging since early 2000, I was really acutely aware of all the pros and cons of the various different methods. I'd worked with many different cameras, you know, single photon detectors, EMCCDs, you know, like that was before CMOS cameras existed. And certainly all of those technologies are getting better and better. But when I saw this paper come out in 2014 by Eric Betzig's team in Science, I was really blown away by the quality of the data that they were showing. And even more so by the fact that they were able to show this in maybe 20 different scenarios where they were looking at live cells. And it was just really striking for me. And I was already aware of light sheet. I'd been to the conferences, the light sheet microscopy conferences, and I always attended, you know, the seeing is believing, and what else? The other one, that the FOM I always attended, focus on microscopy. So I sort of was becoming aware of light sheet. But for those of you who may not be aware, light sheet microscopy is distinctly different to confocal and wide field in that it illuminates, you know, in the early iterations of light sheet, there were two objectives. There was one objective that would deliver a sheet of light to the specimen. And then there was another objective that was placed orthogonal to that, that would detect whatever focal plane was being illuminated. And then that illumination can either drive up and down through the specimen, or in some cases, the specimen is driven through the light sheet. But the big advantage is that you don't illuminate any areas outside of what you're actually detecting with your camera. So that is your focal plane. And so this means it's much less phototoxic. It's a very gentle way to image cells. And the other advantage is that you get very good signal to noise as well. So remembering back at the confocal data that I showed you how noisy it was, you know, this data was very clean and had a very good signal to noise ratio. So when we saw that in 2014, I got really excited, and we contacted Eric Betzig, and we just asked him if he would help us to build one at WeHi. And, you know, I actually asked Alan Kalman to email him, because Alan was a Howard Hughes fellow, and I thought he would respond to Alan faster, and he had just obtained the sort of Nobel Prize. So I thought he must be getting bombarded with lots of emails. So Alan sent him an email, and you know, Eric enthusiastically sent a reply the very next morning saying that he'd be really keen to try and help us. And at that time he said, you know, there's 3 ways that you can get access to Lattice Light Sheet. One way would be for you to come to Genalia and use our system that we have there in the Advanced Imaging Centre that is run by Leon. And the other way would be to build one yourselves, in which case they were sharing very generously the plans and whatever else you needed in order to be able to do that. And the third way was to purchase it from 3i. So we decided, firstly, we wanted to go and visit Genalia. We couldn't take our Malaria there, obviously, for obvious reasons. And so we went there together with Kate MacArthur instead, and we looked at mitochondrial DNA release. I'm going to tell you about that next. But this, you know, after that, with the help of Eric and his team and the AIC, I should definitely mention, because they were really fantastic in this. We recruited Niall Gagan from the University of Glasgow, who had just finished his PhD, and him and Lockie, who's a physicist in my team, then built this system. So this is just before we finished it here, but we ended up with 2 cameras, and you know, the finished product looked a bit different. But these are some of the first images that we captured here. So the first experiments that we did, obviously, was we labeled our red blood cells with a very nice red blood cell membrane stain, which is difluoronet. And we DHQ, and then we labeled our parasites. So here I think they're labeled with mitotracker, and then we succeeded to image invasion. So if I just take you back here again and show you, you know, this degree of detail compared to the data I showed you previously, which was just wide field 2D DIC. We really got an enormous amount of detail. So here you see the parasite invading, the membrane is wrapping around the parasite as it becomes internalized, and then after internalization, it seals up behind the parasite. So we've never seen this in real time before. This one here, we sort of call the cookie monster. There are 2 parasites invading. So you see they're both in blue here, the parasites, and then you see the membrane wrapping the parasite as it pushes its way in, becomes internalized, and then the membrane seals back up behind the parasite. And so I should mention that up until that point, it was still a little bit controversial as to what was creating the parasitophilus vacuole membrane. Some people suggested, or some previous work had suggested, that it was due to some of the contents of the rock trees, so these lipids that are ejected from the rock tree organelles, and others had suggested that it was the red blood cell membrane that was wrapping around the parasite, but it hadn't yet been fully determined, because all they had to go on was really the fixed data, the electron microscopy data that gave fantastic resolution, and also the other fixed cell experiments that were being undertaken. And as far as the DIC time-lapse experiments, they were able to get really great detail of those early stages of invasion, the pre-invasion stages where the parasite interacts with the membrane. But once the parasite started to be internalized, it really wasn't possible to see, did the parasite actually go in and stay in there? You know, how did the membrane seal up behind it? Those kind of details just weren't visible. So this is also, you know, the big advantage of Lattice to as a light sheet approach, because I know that there are now other subcellular light sheet approaches that are similar to this one. But here you can take any angle through that volume, because these are whole cell volume images, and you can take a slice and you can look at that with detail. So you can see here, this is a section through this volume here showing now these parasites that are now in yellow. and how they're coming in and how they're being wrapped by the membrane. So it was really quite extraordinary, the kind of detail that we were able to get. We also were able to come up with a protocol to be able to quantify then the kinetics of invasion, which you can see here. And here you can see where we're just using a MRS here, applying surfaces onto the red cell and onto the parasite. And then we have a pipeline that allows us to see when the parasite makes contact with the red cell membrane, it turns yellow, as you can see here. And we can export numbers associated with that. So we can generate these curves. And there was another one here. So this is an example of where there's no invasion. So you see the parasites interacting with the membrane, but they actually don't get internalized. And so this is the kind of plot that we can generate. So we're in the process of trying to automate this as much as we can. And then the other thing I mentioned that, it wasn't yet really determined, is the parasitophus vacuole membrane formed by the red blood cell membrane itself? And presumably this can be, the composition of that membrane may change with some of the contents of the rupture organelles that are released. But here, Lockie came up with Lachlan Whitehead, who's a physicist in my team. He found just around the same time that we started to do this work, this method here called LimeSeg was published. And what that allows you to do is draw a circle on the cell of interest, and then it goes in and it identifies the boundaries of the cell. It forms a meshwork on the surface of the cell, which you can see here that is being formed during the time lapse. And then from that, we were able to obtain things like measurements of volume and surface area. So from that, Lockie and Niall then were able to see that during invasion, you get this drop in surface area of the red blood cell membrane. This leads to an increase in reduced volume. So that's more linked to the sphericity of the spherical nature of the cell. And then you can see all of the invasion events that we looked at. And each time you get a drop in the surface area, it's very similar to the surface area for the parasitophus vacuole membrane. So this tells us really that most of the parasitophus vacuole membranes form from the red blood cell membrane itself. So we also did similar experiments as Paul Gilson's group. And here you see just another angle again. So you see these different views, X, Y. Here you see X, Z, and YZ angles. And what you can see is as the parasite goes in, you'll see here it interacts. So if I just take it back there, and you'll see that it causes this large deformation on the membrane, and the deformations are quite striking. They almost get completely wrapped during these deformations. And you can see there's a calcium flux at the apical region, and then eventually there's the calcium flux there. We see this recoil of the parasite, which is typical, and then it becomes internalized. So I wanted to point out too that before I mentioned that with the DIC in 2D, they were only really identifying probably 50% of those events. With this approach, we were identifying more than 90% of the calcium fluxes. And in those cases where we didn't see the calcium flux, we assumed that it was just out of the field of view that we were imaging because we could see that from the data. So we believe that, and more recently we've done these experiments again, and we detected 100% of the time. So it's really, really shows you how just getting all of that information back, the whole cell volumes with good temporal resolution, we can really capture everything. This is just, I won't spend too much time on this, but we looked at different inhibitors, the same ones that Jake and David Rigler looked at. You see this little sort of filopodia that come out when the parasite is attaching to the membrane. We were able to identify a lot of details like that and then describe those kind of things. But I won't go into more detail than that. We also looked at the curvature of the membrane, the negative Gaussian curvature. You can see that when the tight junction is formed, we found that you have this very high level of Gaussian negative curvature here at the internalization site, whereas when you block tight junction formation, you don't get the same degree of curvature. So we start to be able to look at different biophysical properties. Now, I know that it's almost 40 minutes past 12, so I'm going to fly through the next section. But just to say that this is the work that we did at Genalia when we were first looking at the lattice light sheet and evaluating it for our core. And this is work with Kate MacArthur and Ben Kyle. And they had identified in previous work that they did that during apoptosis, mitochondrial DNA somehow stimulates the C-gas, the STING pathway, which leads to the formation of, leads to stimulation of interferon and the stimulation of inflammatory pathways. But they couldn't understand how on earth the mitochondrial DNA was getting out and stimulating this pathway inside of the cytosol. So Kate came to me, and around the time that Eric's paper was published, and she wanted to have a look at that. And we started out by just using our resonance scanner, because that's what the best tool we had at hand at that time. And we used Pico Green, which allows you to highlight mitochondrial DNA. And she also developed this fusion between TOM20, a protein in the outer mitochondrial membrane. With that, she induced apoptosis and looked at whether or not we could see mitochondrial DNA release. If you look closely, and you probably won't really notice, but we did eventually see, when we went back to this data, we could actually see it, but we just didn't believe it at the time, and we just didn't really know what we were looking for. And you can see how noisy the data is. So when we first went to Genalia with Kate, we took the same cells, and this was some of the first data that came off the lattice. And you can see here these mitochondrial DNA popping out. So during apoptosis, the mitochondria undergo, they round up, but they change morphology, they fragment. And then eventually there's mitochondrial damage due to Bax and BAC, and then the mitochondrial DNA is released and it just pops out. So the AIC advised us to use better fluorophores that were brighter and more photostable. So then we went back to Genalia after Kate had generated these new cell lines, and now you see these new cell lines that are instead using M-neon green, which is a much brighter fluorescent protein and much more photostable. And also the halo tag, sorry, that's spelled incorrectly there, but the halo tag and all of these dyes from Luke Lavis, who's at Genalia, which are very photostable and very bright. So these are in control cells. You can image for up to 2 hours on the lattice, continuously acquiring volumes as you go, and the cells can withstand it. And then in this case here, we add in this drug, which induces apoptosis. We also add in a caspase inhibitor to stop them from balling up and flying off. And then if I show you what happens with these ones now, when they undergo apoptosis in this condition, you see that they round up, they start to fragment, and the mitochondrial DNA pops out, which is these little green blobs here. So we were able to quantify that process and correlate it to things like cytochrome C release. Kate then went a step further. She fused mRuby2 to Bax, which is a protein in the cytosol that then translocates to the outer mitochondrial membrane during apoptosis. And you see it's in the cytosol, translocates to outer mitochondrial membrane where it forms these puncta. And then what she found was that the mitochondrial DNA was popping out through these same regions where the puncta were. So then we did in parallel 3D sim, live 3D sim. So you can see the beautiful resolution we obtained, because remembering that lattice is still a diffraction limited technique. And then this is live 3D sim again. And what Kate found was that the Bax rings are formed, these macropores on the outer mitochondrial membrane. And you can actually see that the mitochondrial DNA is coming out through those pores. So it was really beautiful. Like when we went to Genalia, we used both systems in parallel, actually. They were very generous. Kate was running the lattice, I was running the live 3D sim platform, and it was pretty awesome. It was really like, for me, a bit of a Disneyland of microscopy. This is I've got 2 slides to go. I'm almost finished. I just wanted to tell you about, because, you know, this is really all about 4D imaging and why it's really beneficial. But this is a study that we did together with Danny Ratnayake and Peter Curry at Monash University. And they came to us because we had the Zeiss light sheet platform, and we had like the AIRI scan at that time, the 880, and also a multi-photon platform sort of nearby each other. And Danny brought her fish in. Here you see that the muscle cells are stained in magenta. And with GFP, and that's in magenta. And then she also had these macrophages that are stained that are labeled with M cherry in yellow here. So with the multi-photon laser, Verena, who's a member of my team, worked closely with Danny. They induced a wound in the muscle using the multi-photon laser. And then at that moment, you get these macrophages that are coming into the wound site almost instantly. And what they found is that, you know, some of them kind of hang around, some of them are transient. They also then induced the muscle injury with the multi-photon laser and took the fish to the light sheet platform and had a look at it across the whole organism to see what was going on. And there they were able to deduce that there was a subset of macrophages that dwell in the wound site. And then there were others that would come in and go out again. So they were more like a transient population. And they did this by doing some retrospective tracking of those macrophages. And then I think this is my last slide, but they then decided to take the fish to the AIRI scan, and they did this time-lapse recording. And what they found was that these dwelling macrophages were actually sort of, interacting very intimately with the muscle stem cells that are labeled in with the cyan. They're labeled with GFP. So PAX3A is a transcription factor for muscle stem cells. So the macrophages you see there, they have these lovely processes, and they're sort of interacting and cuddling the muscle stem cells. And then have a look what happens here. After a certain time, those muscle stem cells divided. So this led to Peter Curry and his team and Danny doing further studies where they did some single-cell RNA-seq, and then they identified what that subset of macrophage population was, and from there they identified this NUMT, which is a protein that is released by macrophages that actually stimulates muscle stem cell proliferation. So it was really, really awesome study, actually. This credit to Peter Curry and Danny Ratnayake at Monash University, but beautiful piece of work. So I'm going to finish there. Sorry, I took a bit longer than I should have. Just to say a big thank you to my team. This is my team here. Some of them are missing, some of them have left, and I've had a couple of new people join recently. But it's a really fantastic group of people. We're a very multidisciplinary team. Niall is an engineer. He's the one who built the lattice light sheet. He comes from the University of Glasgow. Verena, originally from the Max Planck. She's a neuroscientist by background, but does the most awesome tissue and whole organism light sheet imaging you've ever seen. So if anyone ever wants to reach out to her, she does some really fantastic work. I've now got another engineer PhD student who's working with microfluidics. Mike is a physicist from the US. He came out of Jörg Bühlersdorf's lab, and Samuel Hess. Lockie, physicist from University of Melbourne. Nina, computer scientist, AI specialist. Students, Sabrina is working on spatial transcriptomics, and Cindy is a physicist as well. And Cindy has become a biologist, actually. So she came to me as a physics student, and most of the work I showed you on malaria was done by Cindy and Niall, and they really did an awesome job. And also shout out to Alan Cowman and his team, who have worked very closely with us. John Heddleston and Leong at the Advanced Imaging Centre, and Marte Bureau and Darian Camp at University of New South Wales, and Jake Baum and Dave Rigler, who I worked closely with for many years and inspired a lot of the developments we subsequently made. And Kate MacArthur, who's now at Monash. So with that, I'll say thank you, and apologies for running a bit over time.
Thank you very much, Kelly. Especially, it's actually 1 o'clock now, I think, where you are, 1 o'clock in the morning. So
it's 12:50, I think. Wow, it's 10 to 1.
I'm getting a talk, actually. Actually, my first degree is in parasitology, and then I went into imaging. I imaged Malaria, so close parasite to malaria. So I know.
Oh, really?
So I was reminded of this. So we did some tubulant staining of the invasion, actually, like 100 years ago. I feel like.
Oh, wow! Maybe we should try that again
to see you.
So Kelly, if you stop sharing, I can put the quiz up actually.
Okay, sure.
Yeah, so we give you a very short break, not long, so you don't fall asleep. Just we do the talks so people get a bit more a review of the kind of talk and have a bit of fun with the Mentimeter.
I hope the questions are okay. They might.
Yeah, I'm sure there will be. I mean, they're a little revision of what you have spoken about and give people a bit of a easy way of asking. I mean, just, you know, we can talk it through as well. So it's not.
All right. So here's the first page anyway. Well, we're waiting for people to join. Got 6 people in already. Okay. Sorry about the dreadful pun.
Like you should have said like staying awake.
Staying awake.
Yes.
I have to say,
I don't usually have a croaky voice, but my voice is really croaky now.
No, it was very good. That was lovely.
There we go. We got quite a few contestants. Shall we get going?
Sure.
Sure. Let's move on then. All right. Okay.
So I don't know if you said, but the winner of the quiz wins Foldscope. So that's the tradition, because we started in lockdown and the idea was that people at least would have a Foldscope to do microscopy, and now it's become this kind of tradition. And we send them actually out to people wherever they are. So that's, I used to do it for my village post office, sending out Foldscopes to all over the world in lockdown.
First questions here. What year was the first live cell microscopy of malaria parasite invasion captured? 55, 75, 86, or 48? I definitely heard the answer to this one. And, oh yeah, 6 people got that one. All right. Let's see where we are with the leaderboard.
Good.
Oh, who's up there?
Oh, wow.
Rob is the winner. Right, so far, let's see what happens next. And the question is, oops, enter. What makes live cell microscopy of malaria parasite invasion of cells so challenging? And is it the parasites are only 1 or 2 microns in size? The parasite egress and invasion is stochastic. progress of internalization is rapid and occurs in 10 to 20 seconds, or the plasmodium is very sensitive to light, or all of the above. Okay, we've got 6 people, all of the above, that's right. So the leaderboard. Oh, just a minute. New winner, I think.
Yeah, so, but that's still open.
Emma is in charge.
Rosie. But it's Rosie Manza from CI, kind of the second one in here.
Oh yeah, that might well be true.
Giving away the secret.
All right, third question. Which approach enables us to visualize and study the complete domains of back and backs in mitochondrial? So this was bright fluorophores, low power or resonance scanning, bright fluorophores, a lattice light sheet, a short acquisition, bright fluorophores, resonance scanning and short acquisition, bright fluorophores, lattice light sheet and low power, long acquisition. I can't even read them out fast enough. And
sorry, that was too long.
The lattice.
Of the problems when we tell people to give us questions and answers, if they don't know the ropes, then sometimes these question and answers are a little bit ridiculous.
Okay, so we've got 2.
Just a bit of fun.
Emma is still in the lead. Right. Question 4. Right, so we definitely got this one as well. Can you describe which cell type interacts with muscle stem cells during muscle injury? As B cells, macrophages, megakaryocytes, cytotoxic T cells, or muscle generating epithelial cells? And the answer is macrophages, which was shown in the video. Everyone got that one, right? Obviously, the videos were fantastic. Just a minute, I think. Emma still gets the win. I should look far ahead. Right, last question. Which of the following methods apply to improve live cell imaging? So that's restricting illumination to the focal plane only, undersampling or binning pixels, use of highly sensitive cameras, a spatial and temporal resolution that is compatible with the preservation of cell biology, or all of the above. All of the above. All right.
It's
So
often
now
a good guess, but
the drum roll. Let's see who won. Dun. Oh.
Emma did very well, actually.
Emma,
Yeah, she did. Yeah.
Just identify yourself by putting in a message in the chat. That would be great. Okay, right. I'll hand over to you, Stephanie, for the questions.
Yeah, thank you very much. So this was very nice. There were some questions about the now serious questions. Actually, you could unmute yourself if you're here as well. There's a question about the lattice light, and people were really astounded and found it amazing. But so the question is, you see it maybe yourself in the chat, actually, Kelly, but what is the potential to screen drug and to see if parasite invasion is inhibited, frustrated change of dynamics? So I guess the question is, how much can you multiplex this already?
Yeah, really good question. And apologies to Nick, actually, because I did promise to try and put in a slide or two on our Zeiss lattice light sheet. So I didn't mention that because I only showed you the home-built system that we have. So one of the big challenges with the home-built system is that I didn't really show you, but the biology that we image is on like a tiny little 5mm cover slip. And the objectives are in the upright position. So you load the cover slip with the cells that you're interested in imaging, or whatever specimen you have, into the chamber that's full of media, and then you image from there. So we found it really, really challenging to capture a large number of invasion events, or even like when we're looking at things like cell division. You know, it took quite a long time to be able to find the field that we needed to be in to see successfully cell division occurring. And there was many other examples of that. So we now have a lattice light sheet from Zeiss, the Zeiss lattice, and this has completely changed how, the kind of experiments that we're now capable of doing. And we can certainly image things in a much more high throughput way. We're able to see a lot more invasions. We're able to easily image many cells dividing in the field, and we just have heaps more capacity to do that. And the other thing that they've introduced recently is an ability for us to be able to move to different positions. And yeah, so I would say definitely much more amenable to drug screening now than what the home-built system is. It doesn't quite give you exactly the same degree of resolution that the home-built system has, but it's still very, very good. And most of our experiments now are done on the Zeiss lattice system rather than on the home-built system. So we reserve the home-built system just for some very specialized experiments, but otherwise everything we do now is on the Zeiss lattice. And it's a really simple to use walk-up system. I think, do you have one, Stephanie? Yeah,
No.
but I would say that it certainly has made live cell imaging a lot more easier, and we expect that it will be possible to screen in a 4-dimensional way. The biggest challenge we have right now is that we have huge data bottlenecks. So the pipelines for the data processing and analysis are not well established yet. So that's where we're putting most of our efforts.
So there's quite a bit of effort going into the smart imaging. So you have feedback loops, so you actually understand which cells are worth imaging. And also I think what I saw, the sample preparation is a conventional slides, or
yes, that's right. Yeah,
also because that used to be very inhibitive.
Yeah, that's right. Yeah. So previously it was difficult, and it wasn't really easy to work with in sterile conditions. But now it's an open top system where we can apply CO2 and proper humidified conditions, and we can do really long-term imaging. And we can use different types of formats of dishes. So it's really great. It's an inverted platform, Zeiss, so it's much better.
I've only seen the video, but we're all very jealous. No, I mean, it's great. Yeah, it's nice to see the changes as well, because initially the commercial systems were very close to the in the lab produced systems, which make it actually then, I think then people who developed systems had actually the advantage of high resolution, and now actually making them into robust systems, it usually takes actually one generation of circle of commercialization, I find.
Yeah.
I mean, that's just my opinion, but yeah, that's
but it's true what you say, I think. But I have to say that live cell imaging compared to, because I've been doing live cell imaging over 20 years, as I mentioned, and I really have to say that it's really so much easier to do it now. And there's also a great range of different fluorescent probes available, apart from some very good fluorescent proteins that you can tag your protein of interest with. There is also some really fantastic dyes out there, cell permeable dyes now that bind things like actin and microtubules and DNA, and yeah, there's lots to work with.
Someone was actually Emma, who may have won the quiz, I'm not sure, but Emma is asking if genetically encoded chlorophylls would actually be bright enough to image with that lattice light sheet during the malaria invasion, if that's the case already or will be in the future. What's your
Yes, they are. So we have, Alan's team have established one parasite line, which is M neon green fused to ROM3, which is a protein from the rock trees, I think, from rock tree bulbs maybe. And this is protein is released during invasion, and it's definitely bright enough for us to image. It's very bright. And now there's this fluorescent protein that Nathan Shaner has published that I think he isolated from a jellyfish in up in the north of Australia on one of those beautiful islands, and it's very similar to the Aquarin GFP, but it's much brighter. It's brighter even than M neon green, and so he's, I think this is the next one to take a look at. It's been very promising for that stuff.
Yeah, thank you. And someone is also asking biological question. The damaged mitochondrial DNA will be secreted to the extracellular matrix. Is the question?
Yeah. So the thing is that it's secreted. It's released into the cytosol of cells that are undergoing stress. So when the back apoptotic program is initiated, then this causes damage to the outer mitochondrial membrane, and from there the mitochondrial DNA gets released into the cytosol. And in the cytosol you have these sting proteins that actually are normally residing in the ER, I think, but during stress they come out and they can bind the mitochondrial DNA, and then this stimulates production of interferon and inflammatory pathways. So I don't know about extracellular matrix, but certainly nuclear DNA may do that. But yeah, I think the main pathways identified up to now are related to cytosolic proteins.
Oh, thank you. And Kirti is just making, I think, half a comment, half a question. He seems saying that he seems a bit jealous because he says funding seems to be great where you are and you have all cutting edge microscopes. And he would like to know how you got them, I think. He says, any thoughts, but maybe that's how you got the microscopes.
It's a good question. It's really hard to get funding for equipment in Australia. So most of this equipment was funded, and I realized that I didn't put it onto my slide, but it was mostly came through donations from, philanthropic funding or from donors or from the Institute itself. But, there's not a lot of options for us in Australia to apply for equipment funding. So that's really how we are able to fund it.
Yeah, that's great. Thank you. So you have a sticker on the microscope. It's the fundus names letters light sheet microscope. to remind people that people are donating, and that's how you fund research. I mean, I worked for Cancer Research UK where we did the same because people just want to kind of support, and that's a great way.
Yeah, exactly. Sometimes, I think one of the big funders that we had, I think they decided to remain anonymous, but otherwise, yeah, we put their names on the microscopes.
Thank you very much. Actually, we're now over time, and I mean, I do realize now it's really 1:06, and here you are. So I hope you can have some kind of nightcap tea now, and you can go straight to bed and don't dream about these images. But I think we all will be, because it was fascinating. It was just wonderful to kind of see your exciting work as well as your thoughts, and I mean the way you image and you think about imaging. I think it's just been totally inspirational for anybody, and seeing these kind of applications is just lovely. So